Okay, thank you all for coming to our Mycotoxin Symposium this afternoon. My name is Mike Lilburn. I retired from Ohio State University 5 years ago. The last 2 years I've had the pleasure of consulting for Innovad in the mycotoxin arena. One of the things I realized is being an academic, I've spent a lot of time in the last 2 years in the literature learning about mycotoxins. And what I've realized, because I also spend a lot of time in the field, is the real dearth of appreciation for our mycotoxin status in the US, both in terms of awareness, in terms of mitigation, and some of the neat things that people are doing research-wise. So when I was putting this together, most of the research is being done by companies in the industry and our ARS, USDA group in Athens, Georgia. There's really not much mycotoxin research being done in the US. Certainly quite a bit in Europe, and I'll be discussing just a little bit of that at the end of my presentation. So what I want to talk about this morning is just to give you an overview of mycotoxins from the perspective that what most of us don't think about in terms of not so much the history of it, but how mycotoxins became relevant, but also some of the importance of mycotoxin management before we actually have an ingredient or a complete feed. So again, these are our 4 classes of typical mycotoxins: the fumonisin, the oxynavalinol, DON, aflatoxin, zearalenone, and ochratoxin. And I have this quote from Fisher in 2012. Fungi are proposed to be the greatest threat to animal and plant health among all the taxonomic classes of pathogens. And I think I was really taken with this quote, and certainly this applies to each harvest year as we go through a differential series of circumstances before we get our grains and our crops. So again, being an academic, I spend a lot of time trying to learn from the literature. And in my next slide, I'll be showing you really the first documentation of actual mycotoxicosis. But I just wanted to point out here that, as early as 1947, and then some reports from the 1950s, people were seeing what we now understand were mycotoxin challenges. But mycotoxins at that point really weren't identified, weren't recognized. But these are some of the first observations that I could find in terms of what we now know as a mycotoxin challenge that was actually recorded. The first actual evidence of mycotoxicosis occurred at approximately the same time in the early 60s. 1,000 turkey poults died after consuming a sterile but toxic peanut meal. And this occurred in the UK. And about the same time, there was a high incidence of liver disease in Kenya, where they were able to isolate high levels of Aspergillus flavus. From ingredients from both of these outbreaks, they were able to isolate toxic fungal metabolites, were extracted and actually purified, and aflatoxin was identified and given the name. Aflatoxin is highly carcinogenic, and so not only were we concerned with the adverse effects from a production standpoint, but also when we found out how carcinogenic was, basically aflatoxin today is the only mycotoxin that is regulated at 20 parts per billion, both in Europe as well as in the US. So you may see recommendations. You may see suggestions for levels of different mycotoxins, but aflatoxin is the only one that is currently regulated, and very tightly. And in my opinion, and I'll be giving you some of Mike's opinions today, you don't have to take them, but in my opinion, really, aflatoxin in commercial grains today is something we're not really concerned with because of this tight regulation, because of its carcinogenicity. In Africa, where peanut meal is used extensively, aflatoxin is reported now to be approximately causing 40% of all the liver cancer in cases in the African continent. And if you think about the developing world, not only is there not the resources, we'll say, within different countries to test for mycotoxins in grains, but really even when you have high levels of given mycotoxins, in this case particularly aflatoxin in peanut meal, the population doesn't have an alternative, to look for other food sources. And so that is why one of the reasons in the developing world where mycotoxins are really, from a human standpoint, still quite a concern because of the resources and the choices people have available for the foods and particularly the grains they eat. So what I want to talk about here is just to give you a little bit of background on the mitigation of mycotoxins and make us aware of the fact that a mycotoxin program, mycotoxin management program, is not typically what we think about is, okay, after the grain comes in, it's harvested, it's dried, it becomes an ingredient, okay? What I want to talk about is just to give you a bit of awareness of the knowledge base on things that can be done within a given harvest year to potentially manage the mycotoxin challenge that may occur. You have biological factors, susceptible crops, compatible toxigenic fungi in the soil, environmental factors, temperature, both extremes in temperature and certainly climate change. A number of recent papers in the literature on the effects of climate change on predisposition for aflatoxin development and other mycotoxin developments, mechanical injury, the harvesting, crop maturity. And then finally, as it goes into storage, and some factors during storage that then result in the grain that we're going to be using in our feeds. So an awful lot can be potentially thought about and managed even before the crop goes in the ground. So from a pre-planting standpoint, you can think in terms of the hybrid used, whether it's a short or a long season hybrid. In this case, it would be of corn. Tillage or no tillage. This can have a significant effect on the potential for fungi development within your crop. Crop rotations and planting density. And for each of these, I have references included, and these references will be included in the papers that come out of this symposium. Okay, staying on with pre-planting, tillage, crop rotation, cover crops, that these considerations particularly can influence the incidence of DON, or deoxynivalenol. And certainly in the last couple of years, this is the predominant mycotoxin that we're observing in the upper Midwest. But again, tillage, crop rotation, cover crops are all factors during pre-planting that can mitigate fusarium development and DON in the grain. Crop residues facilitate overwintering survival, potential dispersal of fusarium, and even when minimum tillage would bury the fungi. So in complete no-till systems, we could be very much predisposed, depending on the other factors of environment. And again, fusarium head blight in wheat. Wheat is the one that, particularly in Europe, there's been an awful lot of work done with wheat because it's the predominant grain in Europe. And DON contamination can be reduced if, for instance, wheat does not follow corn and crop residues are buried through tillage. So here, these are just some factors in terms of how we're managing our crops, crop rotations, that could potentially minimize the incidence of fungus and ultimately mycotoxins. Insect management. And again, a lot of our crop varieties, and Tony Glenn from ARS is a plant pathologist, geneticist, and he may be talking a little bit about some of the genetic components of some of our grains that could influence aflatoxins and mycotoxins. But again, BT has been very effective at insect control. Weeds, and the competition of weeds for soil moisture and nutrients. And so particularly in a very, very, very dry year with drought conditions, and you have excessive weeds, in your fields, this could be a predisposing factor, again, for mycotoxin development down the line. And again, field observations, knowing what your crops look like, doing scouting periodically, scouting in relation to insect challenges and things of this sort, to know what to expect at the time of harvest. If you know you have a particularly weedy crop or a lot of insect damage, you know, how do you integrate this into your harvest schedule to try to, again, minimize mycotoxin development? And again, this is a meta-analysis from Pellegrino in 2018 for genetically engineered crops. And so certainly there has been a lot of improvement and potential in using some of the GMO varieties. The meta-analysis results, and this was over a 21-year period. It was evidence that genetically engineered maize performed better in terms of grain yield, higher with lower concentrations of mycotoxins, particularly fumonisin and the trichothecenes. You know, some of these long names you have to have experience in being in this field for a while before the names kind of rattle off your tongue, and I haven't been in the area long enough. But anyway, but there's a potential here again for some of the GMO crops in terms of mycotoxin development. My last couple of slides I want to talk about are emerging mycotoxins. And by emerging, it's not necessarily that these are new mycotoxins that are developing. But emerging really has to do with the technology now involved in mycotoxin development. Certainly between mass spectrometry, ultrasensitive, high-resolution mass spec, we can now actually measure and determine the mycotoxins that we didn't previously know existed. And again, a couple of papers, Street and Gruber-Dominger in Europe in 2017, where they actually can now determine the levels of some of these emerging mycotoxins, okay? And in my last slide, I'll be showing where these are becoming of particular importance because we're now aware that they're there and we have the technologies and the techniques available to measure them. But when I say emerging, again, Joe Jambrone at Auburn had the first paper in the literature on tenoxanic acid, 1978. And so it's not emerging in terms of something new. It's just something now we're aware of because we can detect it. And my last slide, the reason why I think it's particularly important is that tenoxanic acid is really on the radar in Europe. And tenoxanic acid is one of the mycotoxins produced by the Alternaria species of fungi. And many people are not even aware of the Alternaria species. But tenoxanic acid is one of the toxins produced. And in Europe, you can see a number of these references I have here where tenoxanic acid is showing up in baby food and human food. And certainly in, I would say, 90% of the samples that we have analyzed, blood samples through Innovad, I would say 90% of the samples where other mycotoxins are not necessarily either present or at low levels, we see tenoxanic acid. Not necessarily in high levels, but it's consistent and it is persistent. So I'm just, again, from an awareness standpoint, once you start getting some of these mycotoxins in baby foods and the human food chain, it takes on a different degree of interest and attention from governmental regulatory agencies. And I think that's an important part of this symposium this afternoon as you listen to our speakers, is that there are some unique things being done by companies on research. But again, once it gets in the food chain and it gets out there, Pandora's box is open. And we really need to be aware of that. The other thing that's really kind of interesting here, it's the Hagenal paper, is that tenoxanic acid, if in wheat milling, okay? If you take out the flour during wheat milling, there are very, very low levels, if any, tenoxanic acid. It is very, very high where it's present in the byproducts. And so from a practical standpoint, you know, we have wheat byproducts in the US that come into any number of our poultry diets. There's probably not a poultry species that at some point in their life are not fed diets with wheat byproducts, particularly wheat midds. And so it's not necessarily even the grain itself or the intact grain, but it's some of these byproducts that we use extensively. And I think most of us here that are in the production world also understand the concentration effect you get in distillers grains. And so if you have X level of DON in corn, of course that level of DON is going to be exacerbated in distillers grains. So the processing side of it also is of importance. So in summary, mycotoxin mitigation begins with increased awareness of various agronomic factors that are unique to each farm and each harvest year. The utilization of free online in-season and post-harvest crop reports, extensive reports, put out by DSM, by Alltech, by Neogen. They're free. I think, we have access to awareness of what a given crop year is like. And I think we need to avail ourselves of those resources. And the increased prevalence and awareness of emerging mycotoxins should not be ignored, particularly as they relate to human nutrition.